Hi, good morning. I'm Qianling Lin. I'm from Institute of Molecular Biology Academia, Sinica in Taiwan. And today is really my pleasure to talk to PSBMB Young Scientist Forum. I thank the organizer to give me this opportunity to introduce my research as well as IMB. So my research, which I'm going to talk about today, is on RNA splicing. I will explain to you how the branch site choice affect RNA splicing. So let's start from the significance of post-transcriptional regulation. We all know the gene expression starts from transcription, and after pre-mRNA is made, the introns will be removed and exon will be ligated through a process called splicing. After splicing, the mRNA is exported out into the cytoplasm, and there they are facing the regulation of RNA stability, localization, and translation, which we don't have time to talk about today. So today we'll focus on the first processing. And splicing is regulated by three key elements in the introns. One is 5' splice site at the 5' of the intron, and the 3' splice site at the 3' of the intron. And very close to the 3' splice site, there is a branch site, branch point sequence or branch site. And in between branch site and 3' splice site, there's often a polypyridine track, a UC reach track. Okay, and all three of them are very important for the U2 or the U2 auxiliary factor to recognize. So again, if we zoom out, we see that the U1, the small RNA in the U1 will base pair with the 5' splice site, and the small RNA in the U2, which is shown by the sequence here, base pair with the branch site sequence as shown here, okay? And at this stage, this pre-spliceosome already determine where the splice sites are. And I will not go into the detail of the reactions later, but basically U2 is the one that determines the 3' splice site, but it does not base pair with the 3' splice site itself. Instead, it base pair with the branch site sequence. So the branch site sequence is very important. Biochemically, it's also very important. We know, as shown in the picture before, the U2 sequence recognize the branch site sequence and push out one nucleotide we call branch point. And this bulge nucleotide will perform nucleophilic attack on the 5' splice site, forming the lariat intermediate. And now the first exon has a free 3' end. So now the 3' OH will nucleophilic attack on the 3' splice site, forming the ligated exon and the free intron. And if the branch point really perfectly base pair with the U2, we should have a very canonical branch site, such as TAC, TAAC in the budding yeast. However, it is not the case in human introns. Human genes have very big introns. It has 10 times bigger introns compared to the exons. And in this 1 to 2 kb intron, we have a very big branch site sequence. The branch point itself is more likely to be an A, and the -2 position is more likely to be a T. Other than that, we don't have a very strong branch site sequence. And myself and many other scientists have spent a lot of energy trying to catch this lariat molecule so that we can detect the linkage between 5' splice site and the downstream intron sequence. So we know where is the branch point. So a lot of efforts has been spent, and the scientists found that the more sequence you get per intron, the more lariats you get per intron, then the more branch points you can detect per intron. So basically it's saying if you sequence deep enough, then ultimately you will get multiple branch sites in almost all human introns. So this is one example. They show in the single intron, they detected more than 10 branch sites. So now we know the branch site is very vague. Almost all human introns contain multiple branch sites. But we also know that the branch point is very important to determine splicing. And dysregulation of branch point will cause aberrant 3' splice site selection in some disease model, which we don't have time to talk about today. So I'm puzzled by this question. Is there any consequence of branch site mutations? Will that be okay to mutate one mutation because we still have a bunch of other branch sites, or some branch sites are really important for splicing? To answer this question, me and Jiang Honglun and Chen Yiting start a massively parallel splicing assay. So the idea of this assay is that we want to collect as many experimental data as possible. So we first collect the branch site mutations that are reported in human disease databases. So these are all natural human disease mutations in SNP database, human gene mutation database, ClinVar, and cancer genomic database. If the mutation fall on the branch point or -2, -3 positions, because they have the highest information of branch site, then we collect them and synthesize them into oligo. We synthesize a part of intron and exon, and we always synthesize them in pair. Every mutation, they will be partnered by a wild type sequence. And we have priming site at the two ends of the oligo, so we can ligate the oligo into a splicing mini gene. So we have a 3 exon splicing mini gene, and in the second splice site, we have our synthesized oligo. So remember here we have 12,000 different sequences, about 5,300 pairs of different sequences here. And we have an intron mutation that fall on either 0 or -2 or -3 position of branch site. And it associates with the exonic barcode so that if the intron is spliced out, we can still detect the genotype. And we sent 12,000 different sequences into human cells for splicing. And they are naturally spliced in the human cell, and we recover the RNA and the library by RT-PCR. So we put primers on the first and second exons and try to PCR them. So if the intron retains, then we can PCR the intron. If it's spliced, then we can PCR this ligated exon. So again, here are pools of 12,000 different sequences. So we send amplicon for deep sequencing. And then we use statistical methods to calculate if a mutation affects splicing. And we found about 6% of mutations, they affect splicing. And most of them downregulate the canonical splicing, so they have negative effect on splicing. And we also noticed that among the strongest mutations, a lot of them, they are novel AG mutations. In other words, these mutations create a novel AG in the intron. So the mutations either affect the choice of canonical splice site or they affect the splicing efficiency. Together, we have about 300 high confident candidates that have over twofold change. We can visualize them in the integrative genome viewer. We see here, for example, a wild type use canonical AG, but the mutant does not. And we also validate them by single transfection and RT-PCR. We validated 39 out of 40 intron pairs. So that suggests our platform is very reliable. So with that massively collected data, we want to see what's the underlying rule of the mutation effects, why we only have 300 mutations significantly affect splicing and not the rest. So we first see that the high confidence significant mutations, they are more likely to be mutations on the branch point 0 position. And the -3 position mutation are more depleted in the highly significant candidates, suggesting the -0 position has strongest effect on splicing. And also, we see this is more likely an A to G mutation. The wild type is more likely to be an A in the significant candidates, and it's more likely to be a mutant G for the significant mutations. We also see that the significant mutations, they are highly conserved, determined by the Philo P100 scores. And we also see that the significant mutations, they are closer to the 3' splice site. If the 0 indicates the 3' splice sites, then we see the significant mutations, their positions shifting to the right. So we have so many features of a significant mutation. We want to build a model to describe all the features and to summarize them. So we perform machine learning on these features. We first separate the AG and the non-AG mutations because we believe the novel AG is a different question. They are two AG competing for splicing, and the non-AG mutations, they are branch site mutations. So we create two models by the GLM model, generalized linear regression, and we use loss-Su variable selection to select for significant features. So the top feature that is been selected through the machine learning method is the novel 3' splice site score, meaning that if I have a novel AG here, then the strongest the splice site is, the more likely I use this AG. And the negative feature we found is the distance. If it's large distance or the canonical 3' splice site is very strong, then it's less likely for the novel AG to be used. And through our modeling, we can achieve a very high prediction rate. We have a high sensitivity and specificity for the prediction. And one thing that I probably don't have time to go through today is that we found the selected feature fits very well with the exon definition hypothesis. This hypothesis is saying that because human intron is very long, so our spliceosome actually first define the exon boundary first. We found a lot of exonic features of this potential exon region. If this region look very much like an exon, and it's very likely this novel AG will be used. And for the non-AG model, we found the mutation G is an important feature, and high conservation also promote the significant splicing change. And the negative feature that suppress the splicing change is the wild type splicing efficiency. So this splice site is strong, then it will suppress the change of splicing. So just to summarize the modeling, we find that for a non-AG mutation, if the splice site is weak, it more likely promote a significant splicing change. And if the wild type and mutant allele has a different branch point prediction, then it's also likely to affect splicing. If it's a G mutation, if it's close to 3' splice site, if it's branch point 0A mutation with high conservation, and it's likely to promote branch site dysfunction. On the other hand, if we have a novel AG in the intron, then what's the feature that promote the novel AG? First, this novel AG is very canonical. It has a polypyrimidine track with a Y in front of AG. It's a very good splice site. And the canonical AG is weak. It's close mutation, and it no longer base pair with U2. And it has the exonic splicing enhancer features that attract the SRF or SNRMP enhancing proteins. or it has a high GC content, which is also a feature of exon. It promotes the usage of novel AG. So with that, I would like to provide more explanation from genotype to phenotype because we have a lot of sequencing, a lot of GWAS data, and we'll always get intronic mutations. And how to assign functionality to these intron mutations has been a very, very big problem. And I hope my massively parallel splicing assay can provide some hints linking the genome variants to the RNA splicing outcome. So I would like to thank my team and our state-of-art core facility, our genomics core, our bioinformatics core. They help us to set up something we were not so familiar with. And I thank our collaborator in the Statistical Science Institute. They supervise our statistical modeling. And now I want to switch gear to introduce our Institute of Molecular Biology. We are one of the best research institutes in Taiwan, and we have now 37 labs. Our director, Chen Huairong, just joined us this year from UC Davis. And we really have state-of-art core facilities, which is very important. No matter what you want to do fax, you want to do imaging, you want to do genomics, you can always find people to help you. And I think that's very important. And our research focus are on chromosome biology, developmental biology, and we have a lot of different model organisms. Zebrafish, you see here, we have mice, we have Drosophila, C. elegans, yeasts, and human. And we have infection and immune biology, we have neuroscience, and we have plant biology and RNA biology, which I myself am involved in. And we have structural biology, we have cryo-EM on campus, so a lot of things you can explore here. And we have system biology, similar to my research. In this area, we try to study a network rather than a single gene. And we also have international graduate program for all the international students. Our institute is one part of the molecular and cell biology program of TIGP, and we accommodate about 8 to 10 international students. For total, we accommodate maybe up to 20 students per year, and they're really brilliant and active young scientists. And we are like a big family here. And one of the very important activity in our institute is the summer internship program. We also welcome international students in summer. If you're curious about a 2-month internship, you should definitely join us because we are not university. We don't have students in the semester. So in summer, we are all very excited to see kids, enthusiastic young scientists. So we are very devoted and we're very excited. And all of our faculty really, really welcome young students. Okay, so thank you very much, and I look forward to learning more from you.